Good afternoon, everyone. It is my absolute pleasure to introduce you all to the next exciting seminar speaker, Edwin Antony. Edwin's going to be talking to us all about recent work that he's done on protein A, replication protein A, which is fascinating work linking protein and DNA interactions. Edwin got his PhD, dare we say how long ago? It's quite not that far back, is it? Yeah, 2005 in Wesleyan in Connecticut, and then postdoc at Johns Hopkins, and then in St. Louis, where he's now based as a professor. We met, I think, online on Twitter, Edwin, a while back, didn't we? Before in person, and Edwin was one of the early adopters of the iFlex, and so we're really excited that he's going to be talking about today some data that he got in his lab on our new instrument, which is just brilliant. So without further ado, I will hand over to Edwin. Edwin, the floor is yours.
Well, thank you, Tim. I appreciate it. I can see a lot of familiar faces or names, at least, on the board, so that's wonderful. Yes, Tim, I'm going to let you moderate that a little bit because I can only see a sub-selection of the people on my screen. So yeah, hi. Okay, so this, since the audience is quite big, what I thought I'd do is to tell you sort of a summary of the work that we've been doing over the last 5 to 7 years on replication protein A. So and in many of the slides, you're going to see some data followed by a model on the left. So I won't have time to go over all the data on the right side, but you can just focus on the model on the left side of data. And I'm happy to hang around and sort of talk about nitty-gritty bits of the data and the details after the talk as well. So before I start, let me say, I hope this works. Yeah, okay, so it works. So let me give credits to the people who did the work. Nilisha and Sahiti were graduate students in my lab who sort of started many of these projects as part of their PhD program that have been now taken over by Vikas and Rahul, who are postdocs and a scientist in my group. A lot of the work on RPA phosphorylation that you'll see was actually done in Professor Origanti's lab by her postdoc Poonam. And a lot of the hydrogen deuterium exchange and cross-linking MS experiments that you're going to see are from Brian Bothner's lab, and of course we have a long-term collaboration with Maria for single molecule studies. There's will be work from other people as well. I'll sort of acknowledge them at the end. All this work that you're going to be seeing here was funded by NIGMS, and who've been kind enough to support our work on RPA. Okay, so this is my favorite sort of slide to start off with. Of course, you got to give hats off to Watson and Craig and Franklin to have solved the structure of DNA. And so in their paper, they sort of suggest, right, so you got to unwind the DNA in order to access the genetic material. And so when you unwind the DNA, you end up with single-stranded DNA, right? And so these single-stranded DNA intermediates are sort of found in all DNA metabolic processes, whether you're replicating the DNA or you're dealing with the telomeres, transcription, HR, mismatch repair, nucleotide excision repair, branch migration, so on and so forth. And in all these cases, you're opening up the DNA, whether you're completely unwinding it or you're opening up into a bubble, or you're making polarity junctions, whatever it be, you're exposing single-stranded DNA. And so the consensus is that single-stranded DNA is a lot more susceptible to be damaged compared to double-stranded DNA. And so you have the cells have evolved single-stranded DNA binding proteins to sort of bind and protect these transiently open single-stranded DNA structures. And so these SSBs come in a variety of shapes and sizes, right? In the phage, it's a monomer. In the case of bacteria, it's mostly a tetramer, and sometimes it's a dimer in Deinococcus radiodurans, for example. So in the eukaryotes, this SSB has evolved to be a heterotrimer, and this is called replication protein A or RPA, right? So, but they all sort of perform the same function. They're supposed to protect the single-stranded DNA from nucleolytic attacks and damage. They serve as a template for enzyme recruitment because they often have protein-protein interaction domains. And all of them are essential proteins, right, because of their role in DNA metabolism. So, and given their uniqueness in these different organisms, right, they're sort of interesting antibacterial drug targets, for example. So, so these proteins are extremely important for function, and they function in many roles. And so if you think about replication protein A, for example, right, almost anybody in DNA repair and recombination and DNA metabolism would have a cartoon of RPA in a model that they draw, right? So the problem comes in as to where do we draw this model of RPA? That is always my most amusing part when I sit in a talk, because sometimes it goes on one strand, but not on the other. Sometimes it's coded by 5, 6 RPA molecules on one versus one of the lagging, one of the leading. Sometimes it's on the replication fork. Sometimes it's on the bubble. Sometimes it's not there at all, right? So it's amusing as to either we do not pay enough attention to RPA's role in these processes, or it is just you sort of think of it as somebody said, I think once, it's a molecular grease, right? So wherever you want to stick it, you stick it on, and that's what it actually does, right? So our take on RPA is, of course, something completely different. We think if you're going to derive models of DNA metabolism, you have to go through RPA, right? And so the reason being something like this, right? So if you were to take a piece of long DNA, RPA is going to be bound on the single-stranded DNA substrate. But the DNA is actually buried under RPA, right? So if you're an incoming enzyme in a pathway, you are not seeing the DNA at all. So how are you getting through RPA in order to get to the single-stranded DNA? Becomes an interesting question in itself, right? And so then there are questions about specificity, right? So how can one protein function so many different pathways, and how is it regulated? If the DNA is buried under RPA, how come an incoming enzyme gains access to that single-stranded DNA? Then there's this problem of affinity, right? Because RPA binds a single-stranded DNA with extremely high affinity. So if an incoming enzyme binds with DNA with weaker affinity, how are you going to remodel or replace RPA in order to sort of get to the DNA? So you have to think about the timing of when RPA gets on there. When do these incoming proteins get on the DNA? RPA also binds a DNA with polarity, right? So these domains in the largest subunits sort of tend to bind towards the 5' side of the DNA. So the incoming enzyme, if it has to sort of access the 5' versus the 3' side of the DNA, you could use RPA as a vehicle to sort of situate yourself appropriately on the DNA, right? And then finally, RPA is heavily modified through acetylation, through phosphorylation, simulation. And so how do these modifications then regulate RPA? So these questions have kept us, in business for, the last 10 years, and I think it's going to keep us in business for another 20 years as we try to figure out, how RPA functions in these many processes. So with that in mind, so let me tell you a little bit about RPA. The sophisticated way of drawing RPA is usually like this. So you put a 70 subunit, a 32 subunit, and a 14 subunit, and so these. So the protein always functions as a constitutive trimer, heterotrimer. And it's made up of, but if you want to functionally think about RPA, you really have to think about it in terms of its DNA binding domains, or DBDs, or protein interaction domains, PIDs, right? And so each of these domains are actually oligosaccharide, oligonucleotide binding domains. And in RPA-70, which is a large subunit, there are 4 of them really. So it's A, B, C, and then this OBF, right? So A and B and C are DNA binding domains, known DNA binding domains, and OBF is thought to promote protein-protein interactions. Then you have RPA-32 subunit that contains one DNA binding domain, DBDD, and there's another protein interaction domain, which is also called the winged helix domain. The final subunit is RPA-14, and that actually also has an OB domain. But whether it binds to DNA or not is sort of controversial. If you separate it by itself, it doesn't seem to engage DNA. But if you're looking at either cross-linking or HDX, there are DNA-induced changes, but we really don't assign a function for DNA binding to this particular domain. So to simplify things, if you're talking about DNA binding domains, you consider A, B, C, and D as the primary DNA binding domains. So we recommend people draw RPA more like this. It will be a step up from drawing it as a 3 subunit complex, because as you can see from the talk, there is a lot of complexity to how this protein actually works. All right, so these protein-protein interactions domains are sort of interesting because OBF, which is on the large subunit, has its own Pandora's box of interacting proteins. And so if you want a more comprehensive updated list, you may want to look at this wonderful review from Maria Spies and Colin, where they sort of update this list. But that's 2020, so it might have changed, over the last 4 years. So similar to OBF and its interacting partners, this winged helix domain has its own set of interacting proteins, right? So this goes back to this idea of if you want to access the 5' versus the 3' side of the DNA, you could potentially interact with either the winged helix domain or the OBF domain to gain access in a differential manner. All right, so a bit of historical context as to why some of the models of RPA are biased in the literature. It sort of comes to assigning binding affinities, right? So back in the day, folks had actually isolated the domains, so DPD-A or B or the AB together, or the trimerization core separately. And so this idea evolved that A, B, or AB together are the high affinity DNA binding domains of RPA. Now the trimerization core was thought to have weaker binding affinity, so it's not. So people think that it actually has a secondary role in DNA binding, right? Sort of engages later, for example. But from our work, we know that the trimerization core in general is not very well behaved in solutions. So a lot of these numbers, KD numbers assigned to the trimerization core should be taken with a grain of salt. So together, they sort of, when all domains are engaged on the DNA, you really form this sort of high affinity complex, right? So the reason we got interested in RPA is because if you look at the structure, this is the trimerization core. It does not move a lot, and it actually has quite a bit of DNA contacts compared to A and B, which tend to be more dynamic because of their linkers that are disordered linkers that are connecting these domains. So we thought, would it be really cool to actually think about RPA and the domains in terms of its dynamics, right? So here is the classical model, right? So you have RPA. If you bring in a piece of DNA shown in blue over here, and these are the domains. And by the way, you're going to be seeing these cartoons over and over again. And so I've sort of labeled the domains according to their colors, so you get some idea behind. So the models are always that if you actually have a piece of DNA binding A and B, because of their high affinity nature, would actually get on the DNA first, followed by the trimerization core. So this particular model where you can actually put the trimerization core first, followed by this complex, is not favored because this domain is not high affinity enough. So we thought, can you actually watch this or actually capture this process, and can you test this model and if this actually is true? And so what we thought would be really neat to do is if you could site specifically label each of these domains, then if you put a fluorophore on A or D, and then you can actually move the corresponding fluorophore on the DNA, right? And then you can actually watch how the DNA is moving with respect to each of the domains. Now, ideally, we'd want to be able to label A, B, C, and D, and be able to do this entire repertoire of these experiments. However, in our way of doing these experiments, A and B were amenable, but you can't touch B or C, and they sort of, they lose about 10 to 20% activity, and so that's something that that won't work for the kind of analysis we are trying to do. So the positions that we have published so far on A and D work really well. The protein almost retains 100% activity, and we cannot see any measurable differences in the way the protein is actually behaving. So we've now made the fluorophores for A and D in the yeast, and we have A and D in the humans, and I'll show you some of this well. So this was sort of the template, but the challenge now is, how do you take a protein that's got roughly 1,200 amino acids and you stick in a site-specific fluorophore without affecting the function, right? That is the challenge. And so we played around with a lot of approaches, but we ended up with the non-canonical amino acids to do this. When we started this work, I was being very ambitious, and my graduate students were being very naive to say no. And so what I told them is like, not only are you going to label these fluorophores, but you're going to position these fluorophores so that when they bind to DNA, you're going to see a change in the fluorescence, right? So you can get a readout as to when this domain sort of engages the DNA. So the challenge essentially compounded quite drastically when we sort of went down this path. So based on what was available in terms of the structure, we look for residues that you could potentially touch. And there's actually a paper that Sahiti published where she actually lists all the residues we actually tried. And you can see our success rate was 1 out of 17 or something like that. So we have one position in D, one position A that we actually touch, and we just got lucky with it. That's about it. So we ended up using unnatural amino acids, so we can actually go in and engineer a site-specific fluorophore into the position that we want. And our amino acid of choice was 4-acetylphenylalanine. And so once you incorporated it, you can actually conjugate the fluorophore using click chemistry. And so this is one of the fluorophores that's really worked really well for us, this MB543. And you can see here, when you put the TAG and the 4-ACP into DBDA, you can get site-specific labeling in A, or if you stick it into D, you can get site-specific labeling in T, right? So we've optimized this quite a bit. We've shared this information with many of you. However, there are issues with taking this particular approach, right? So I can tell you right away, don't send your students running to do these experiments right off the bat. Talk to us if you want to use it. So the overall protein yield using this approach actually drops drastically. So you can have a 60 to 70% loss off the bat. Now, if you're thinking about very beautiful ideas of putting in 2 fluorophores, now add that as another 30% drop on top of this, and it's going to drop even more, right? So you cannot use standard copper chemistry because at least for RPA, it definitely clips the protein. And we've seen this with many other proteins that to a lot of these mammalian or higher eukaryotic proteins. The strain-promoted cycloaddition, with the DBCO works really well, but again, you have another 30 to 80% loss in the labeling efficiency, but that can be optimized. So depending on the positions you are playing around, we've got it up to 80% in some cases, but some cases it's so low that it's not enough, right? And of course, you got to for every single construct, you have to optimize this extensively, right? So that means you got to start from the cell line to the buffers, to the temperature, labeling conditions, everything's. So it's not trivial, and it's also expensive, right? So these are all the drawbacks, that are that's an issue with this particular approach. But if you can pull it off, it actually works quite well. All right. So a successful experiment will look like this, right? So in this case, we have an MB543 probe labeled in position A or on position D. And so here's a stop flow experiment where you take the protein against buffer. You don't see a change in the signal. When you shoot in DNA, you see these captures as well, nice, real robust kinetic change, right? So we have a signal for A that we can use. We can use a signal for D you can use. Again, you can already see the limitations here, right? So the signal for A is very robust and clean and really nice. The one for D is actually much smaller in amplitude, right? So those issues actually. Okay. So we call this an FENCA, fluorescence enhancement through non-canonical amino acids. But if you sort of use the same approach to do FRET studies, it's actually a lot better than trying to get a readout like this. Okay. So here's so what I'm going to do is I'm going to list sort of the major findings as I go through the next series of slides. Each one of that is usually a paper or a subset of papers, but I'm happy to talk about these data as we come back to it. Okay. So the first experiment was to sort of look to see, do we know how these domains sort of engage the DNA, right? So we took our labeled proteins, we mixed in with DNA. Here's a stop flow experiment, and we are using DD35 in this particular case, which should be enough DNA to saturate all the binding domains, and we're adding in increasing amounts of DNA. So what you can see here is the signal sort of changes, but at lower concentrations, you actually see a binding followed by a dissociation of DNA binding domain A. But in the case of DNA binding domain D, that domain just gets on there and does not move at all. So you can get all sorts of rate constants from here. But the take-home message here is that this, along with a lot of cross-linking analysis, HTX, MS analysis, and by chopping the domains, we've sort of captured the diffusion of these on CTRAP experiments. But the take-home message is that we think that the trimerization core is actually the more stable part of the protein. So once it gets on the DNA, there is not a lot of rearrangements happening over there, and it actually provides the stability of RPA on the DNA. In the case of the so-called high-affinity binding domains, this thing is dynamic as hell, and it does not want to sit on the DNA. It just goes up and down, up and down. And so it's interesting because if you were to chop them off, and you take a long piece of DNA, and these were experiments that Sushil did in Tech Chip House Lab. If you take the full-length protein RPA and you stick it on the DNA, you can capture the diffusion constant of RPA on the DNA, right? But when you chop off this domain and allow FAB to sort of run, FAB just goes nuts. It's almost like 10 to 20-fold excessive diffusion, barely binds on the DNA, just wants to just flutter away, right? So we like people to think about RPA more of the tricy gets on there, it's holding the RPA on the DNA, and AB is a lot more accessible. that's not a trivial finding, right? Because if you now think about RPA, let's say on a 3' overhang with the junction sitting over here, now all of a sudden, the way an incoming enzyme can recognize that junction is because of the diffusion of the A and B, not because of the diffusion or the removal of the trimerization core, right? So how RPA is positioned and how these domains move will open up different segments of the DNA for an incoming enzyme. So if you're building models on RPA, please consider using a model like this, which I think would be a lot more appropriate. Now, again, this will change whether you're looking at one RPA molecule on a piece of DNA or multiple RPA molecules on a DNA, right? A lot of our interpretations are for one molecule of RPA. Now, how that works on 2 or 3, or if there is any cooperativity in RPA when it gets modified by phosphorylation, needs to be investigated and applied in order for you to interpret the data correctly. All right, so we think there's a dynamic half and a less dynamic half, and the trimerization code is the less dynamic half of RPA. All right, so then we switched over to the second question, and so this is has always been an interesting one for me, right? I showed you how the F domain of RPA binds to more than 24 proteins, and the winged helix domain actually binds to its own subset of proteins. So if we think about RPA in solution in a cell, are these RPA domains just sort of floating around very happily? So the incoming proteins can bind whenever they want, right? Technically, if a binding domain is open. So we said, no, is that really the case? How is RPA sort of going around? When does RPA decides to bind its protein? So the Lee's group over here at Wuhan had actually identified this chaperone protein called RTD105, and they showed that it actually forms a 1:1 complex with RPA. And they actually showed that RTD105 will bind to RPA in the cytoplasm and actually work to promote its nuclear transfer, right? And so we wondered, okay, what is this RTD105 actually doing, and can it actually bind to RPA and regulate its activities? And so here we took an advantage of our ability to move fluorophores on RPA, and so we put one fret pair on either the A or the D domain of RPA, and the second fluorophore on RTD105, right? So RTD105 is a chaperone. It does not really have a lot of secondary structure to it. And so what we found out was that RTD105 actually bound to RPA, to many domains of RPA, and actually configurationally compacted. So the domains were actually not accessible to either incoming proteins. And so that's how it actually holds RPA in place. So it's not this extended form that's floating around, but it's actually more of a compacted form with all the domains together. And we showed that the RTD105 will actually bind stoichiometrically, but really nice break constants and affinity as well. And you can displace this complex RTD105 when you add DNA to this mixture, and we can actually capture that process actually over here. Right? And what we actually showed was that it will remodel the trimerization core and release it first, but not the AB. It sort of held on to the AB domain. So again, goes back to the idea that the trimerization core actually engages that DNA first in this particular case to remodel this particular complex. So then we found out, okay, well, can RTD105 bound to RPA prevent it from binding to other proteins? And so long story over here, there's a lot of AUC experiments. Essentially what we did is we took RPA, we took RTD105, we took RAD52, which is an RPA interacting protein, and followed its complex formation in the absence and presence of RTD105. And what we found out that is in the absence of DNA, when RPA is actually bound to RTD105, it does not bind to RAD52 at all. And so we cannot form that complex. However, so this interaction is actually blocked. In the presence of DNA, it actually releases this domain, and now in the presence of DNA, you can actually engage this complex, right? So DNA licenses RPA to engage these proteins. So these protein-protein interactions actually occur on the DNA when RPA is actually formed. So this was sort of interesting to us. And so I'll touch upon one point over here. So East RPA is actually more open to begin with, and you need something like a molecular chaperone to hold the domains together. Human RPA, to begin with, actually is in a compacted form. It is actually not released at all. So a lot of times, if you try to capture binding between human RPA and an interacting protein, it's almost hard to do it in solution because it's trapped. But East RPA, on the other hand, because it's actually open, will very, very happily bind to a lot of these proteins. So that's why, if you look back in the literature, a lot of the human interaction proteins are either done with isolated F domain or the AB F domain, which now then opens up of the F domain. So that's something to keep in mind that you're not seeing interaction between human RPA and a protein that you're testing, it's very likely in a compacted form and not available to bind. Okay, so do the control experiment, but you take the F domain or the FAB, that will be a lot more useful to you. Okay, then we sort of found out, okay, if you took RPA and if you actually formed an RPA DNA complex, when an incoming protein comes in, does it displace RPA, or can it actually selectively remodel one of the domains to then access the particular DNA? So we had done these experiments where we had the A and the D, and so here we are actually looking at fluorescence enhancement upon binding to DNA using a turf microscope. And these experiments were done by Colleen Caldwell in Maria Spee's lab. And so what she was able to see is that when RPA bound to the DNA, we can actually capture these states. So here we are watching essentially one domain either rotate or come on and off, but they can actually accommodate different states on the DNA. So we can watch that for DNA binding domain A separately, or we can actually watch it for DNA binding domain D separately. And we can quantitate the states in the single molecule turf experiment, right? So here you can see in the absence of presence of RAD52, these states really don't change for the A domain by itself. However, when Maria looked at the D domain, here all of a sudden the states are actually changing, and one of the states actually completely goes away in the presence of RAD52. So here, RAD52 is coming in and remodeling one of the domains of the trimerization code DNA binding domain D in order to access the DNA and form a complex with RPA, DNA, and RAD52. So this actually was the first evidence that actually showed that you can actually see this and watch this in real time. So my secret hope is that someday we'll actually be able to get a probe for all four DNA, all four domains, so we can actually watch, are the other two domains are actually modified or not. But for now, we only have D or A for us to study. Okay, so this was great. So then we turned on to see, okay, you got these domains, these proteins are accessing it, but in all these DNA repair processes, whether it be replication or mitosis or other steps, RPA is often modified by phosphorylation. And so if you talk to anybody who studies or looks at RPA, everybody's always focused on RPA-32 because there's a small disordered region in the N-terminus of RPA-32, which heavily gets phosphorylated and even hyperphosphorylated. However, there are phosphorylations in the large subunit of RPA that folks haven't paid a lot of attention to. And so that intrigued us because some of these phosphorylation events were happening in the DNA binding domains of RPA-70, the large subunit. Interestingly enough, because RPA has these many DNA binding domains, if you were to modify or mutate one domain and look at single-stranded DNA binding using an MZEV, for example, you will always get high-affinity DNA binding interactions, right? So you would think, oh, the phosphorylation really does not do anything to the DNA binding properties of RPA. It only has something else, right? So that's your automatic interpretation. So I think using a much more higher resolution way of looking at RPA, this assumption actually changes. So Sophia and Poonam, so they had actually looked at RPA and shown that Aurora kinase B phosphorylated RPA in DNA binding domain B at this particular position 384, right? And so what happens when you actually phosphorylate it, you actually have a defect in mitosis. So the cells really can't progress through mitosis. And so we actually made a phosphoserine carrying version of RPA. So here using genetic code expansion, we have engineered a single phosphoserine in this RPA-70 DNA binding domain D, and we purified the protein, and you can see nice incorporation and conformation of that using Western blotting. And so here I'm showing you XLMS, cross-linking mass spec analysis, where you're looking at a rough idea of the configurational positioning of the domains between RPA-70, RPA-32, and RPA-14. The data shown here is sort of a differential cross-linking. So anything that you see with a dotted line is unique to that particular condition, and that goes away when it's actually phosphorylated. So I'm just showing you the data for the DNA-bound versions, and you can see for the wild-type RPA, there's a lot of cross-links that actually occur between the domains and within the domains over here. When you put a single phosphoserine in RPA-17, you lose almost most of it, right? So essentially, RPA goes from being like this to a more open conformation on the particular piece of DNA. So what we went on to show is that this particular activity tends to form higher density RPA filaments. So on the same length of DNA, rather than having 1 or 2 RPA molecules, you can now load 4, 5, or 6. And we think that's because some of these domains have been released, allows more packing of these RPA molecules on this DNA structure. But does it have any functional significance? So if you make a 384 to an A mutant, you essentially stall cells in mitosis, and you get a lot of these lagging chromosomes and chromosomal bridges. These are 2 examples over here to sort of see the kind of defects. And in this particular case, when you phosphorylate DNA binding domain B, this position is actually in the DSS binding motif. And so DSS1 is actually a complex with BRCA2. And so we believe that this phosphorylation actually blocks binding to DSS1 and recruitment of BRCA2 in mitosis, which then leads to suppression of homologous recombination in mitosis. So this is pretty cool. We were very excited by this discovery. Now Sophia's lab is sort of trying to understand this mechanism a little bit better. Okay, so that's all the stuff that's actually published. So I'm going to start talking about some of the unpublished stuff here, and sort of what we are trying to do with the EI flex. And you've seen this a lot if you're an RPA aficionado, right? So there's a structure that was actually solved, and you can see that DNA is supposed to be curved through the structure. But you actually start drawing DNA as a linear molecule with all these domains sort of binding across. And we're a big culprit of this as well, because we like to draw DNA in a straight line. And so I've always had this argument with my students that I think the contour of the DNA around RPA is very different than what we are actually normally assuming by default. And so you would see cartoons that look like this. You would see cartoons that look like this. Sometimes you see cartoons that look multiple RPA. It's all straightened out. Right. So can we really understand how RPA is sort of managed, or what's the contour of the DNA around RPA? So there are 3 structures now available for RPA. Some of them have all the domains. Some of them only trimerization code you can see. There's one from East RPA that's a pri-EM structure from pri-EM structure. And then there's the Uslago-Medes structure that was solved previously. And then now there's a Pyrococcus abscessi structure. But in every single case, if you just take the DNA and overlay it, they sort of form this C-shaped or U-shaped, wrapped structure. And the end-to-end distance in this particular DNA substrate appears to be, 55 angstroms of 5 nanometers. Okay, so can you measure it, right? And so people have done this, right? People have looked at this sort of wrapping patterns, and most often with either using magnetic beads or C-traps, where you take a very long piece of DNA with multiple RPAs are bound. And if you don't apply force, the whole thing will collapse, and if you apply force, it will sort of stretch. But still doesn't address the question as to how is our DNA sort of threading through the protein. And Maria's lab has looked at using end-to-end fluorophores to sort of see, okay, can you get an idea of how this actually moves? And so they've done DNA titrations, DNA lengths. But whatever reason, right? So there is some sort of an extension to your multiple RPA molecule, but still doesn't get to what's happening within the piece of DNA. The closest one that we've seen is this work from Wang et al., where they actually used TIFF microscopy to put fluorophores onto 2 different pieces of the DNA. And they actually captured a bending of the DNA, right? But then they interpreted the data more as, like I told you before, as the AB domain, high-affinity domain sitting over there, and the tri-C sitting in the air, right? And you can see how the DNA is sort of going around over here. So we said, okay, that's again does not give us a consensus of exactly how the DNA is being handled by RPA. And this is another question, right? When you have 5 domains that are binding to DNA, can you loop the DNA between the domains? So that's another question that's been sort of sitting around for a while. So we thought we'll actually use more of a in-solution experiment, and nothing is tethered, and we'll actually just use plain old single-stranded DNA with 2 fluorophores to get to this answer. So this is when we ran into Tim, and Tim suggested that we can actually try his new microscope, which I think is pretty cool. And I was fascinated with this microscope when I first saw it, because for a completely different application, because we use a glove box to study proteins that are oxygen sensitive. So we wanted to get this machine and stick it into the glove box and do the experiments in the absence of oxygen. So we had bought it, and then my postdoc got on it to do some control experiments. I say, I want to do this experiment. And he never gave up the instrument, right? He said, that thing is not going into the glove box. It's going to stay out here, and I'm going to do all these experiments. All right. So I'll walk you through to what we did. So we are measuring, essentially taking single-stranded DNA of different length that are, it's a CY5 and a CY3 at either end. and you're looking at the FRET efficiency as a function of DNA length, and from which you're actually calculating the end-to-end distances. So this is raw data for just free DNA in solution, and it actually follows what you would actually predict a polymer to. And so this is the plot for Kim's instrument over here that we have a picoquant in the department. It's about 4 times more expensive than the EIFlex. So we collected the data both on the EIFlex and the MT200, sort of compare them side to side. And so here's the data that you see on the EIFlex over here, and that's shown in blue. And here's the data from the picoquant in sort of black. And so here is the raw data, and the fits to it as to was the confidence. And you can see that the EIFlex data is absolutely gorgeous. I've shown this data to theoretical people, and I think they couldn't believe how good the data was to the theoretical modeling, right? And so I think you guys have done a great job with this particular instrument. So I'm a proponent of it. Okay, so that's well and good. What happens in the presence of RPA. So when you do the EIFlex measurement, so this is sort of what the data looks like, right? You got many molecules that are in this particular space, and you're counting the FRET efficiencies. So you can see samples going from an unbound state to a bound state, right? So in this case, single-stranded DNA, so it's sort of in different states, and RPA is bound, and it sort of definitely should order and go to a more either a high FRET state or low FRET states. If it is fully linear like this, then you would actually go to a low FRET state, and if it's wrapping or something like that, you would actually get to a high FRET state. We also made sure that under all of our conditions that we're using stoichiometric amounts of RPA to DNA, so you're getting one RPA molecule bound to one piece of DNA, and we've sort of confirmed this using mass spectrometry and AUC analysis, and we put a lot of work into identifying conditions under which this happens. Okay, so all the raw data, when you actually plot it into a single plot, this is really what it looks like. We are plotting the end-to-end distance as a function of DNA length. Let me just walk you through it. There's a lot of points over here. So shown in open circles here is just the free DNA that's actually measured in solution, and you can see it actually follows a nice pattern. This is the computational prediction if the DNA were to be completely extended end-to-end, like a linear model like we draw. And so that's what that looks like. So what you see for DNA in solution as a polymer, it's not lengthened up. Clearly, it's in a different amount of polymeric states. Okay, great. Now, when you add RPA, what happens is you see a shift in this end-to-end distance, but this end-to-end distance changes as a function of length, but the amount it shifts, that's sort of plotted over here, does not change by more than 3 nanometers, right, or 30 angstroms. So that tells you that immaterial of the length of the DNA, the contour that it actually falls or takes within this RPA molecules is exactly the same, right? So it says that the DNA that's bound on RPA retains a fluid polymer-like property. There is definitely no evidence of spontaneous single-stranded DNA looping between the different domains of RPA. And the data actually agrees with the partial wrapping model that we see in the structures. So I think you could potentially think about a state where it's going from on and off, but I think around the trimerization core, the DNA is definitely wrapped like we see in the structure. We actually confirmed these findings that we measured in the EIFlex with using deer spectroscopy with Razor. That's one in our lab. So here you're actually putting 2 spin probes at the end of the DNA, and you're adding in plus minus RP and calculating the end-to-end distances. And it absolutely agrees spot on with what is actually measured in solution using this sort of single molecule FRET approach. Both yeast and human RPA, at least for the one condition that we tested, appear to have similar end-to-end distances, but we have to do the entire length dependence to fully understand if there are any differences between the two. So for now, this is what we believe this complex actually looks like. So I wanted some sort of MD simulation to actually capture these biophysical principles for an audience. So we reached out to Yakov and Gargi to try some MD simulations. So I'm going to show you 3 MD simulations over here. So we've sort of fixed the RPA in place. These are coarse grain simulations, and we're looking at the single-stranded DNA in here. So we did a single subunit of single trimer of RPA, or 2 trimers of RPA with 2 different lengths of the DNA. So here's really what it looks like. As we can get to, okay. And so you can see that the overall shape and the contour of the DNA within the RPA molecule does not change, right? So ends, of course, are fluctuating. So, and you can't really pull the DNA between the molecules of RPA. And when you have two RPAs, this DNA you could potentially access to by diffusing the RPA molecule. So, so I think when we draw these models, we should probably put a U shape of RPA in there, which would probably be more appropriate of our interpretation of these models, right? Okay. So why is this important, right? Why is this important? Why is navigating and establishing this path of DNA around RPA important? We had always considered context of DNA, right? So if you have single-stranded DNA in a 3' overhang versus a 5' overhang, versus if you have a bubble or a gap in the DNA, how would RPA actually engage these DNA molecules? So we had collected these data where we could actually move the fluorophore in different domains of RPA, either A or D, or position the second fluorophore either at the junction or at the end of the DNA, right? And we did the experiment where we're looking at either the 5' overhang or the 3' overhang, right? And for a long time, actually, the data did not make a lot of sense because we were drawing the DNA in a very straight line and hoping to understand what it is. And I shelved the data because I couldn't make any sense of it. So until we did these experiments, and now we know that the DNA is actually moving back. And so now these FRET data actually make a lot more sense. So I'm just going to focus on the cartoons over here. What we are finding is that when you actually have a 5' overhang DNA, the trimerization code prefers to stay at the junction. Now, we don't know how it does it, but it always wants to stay at the junction. And we've done this as a function of increasing length, but it still stays at the junction. When you're looking at the 3' end of the DNA, the RPA now diffuses a lot more. It's able to sense many of the domains, and it's actually doing this using the trimerization code, not the AB domain. The AB domain is just going up and down, and so this idea that you can access these DNAs, the junction, when you have a 3' overhang with RPA, is a lot more pronounced compared to when you actually have a 5' overhang. So now I don't know the complete biological relevance of this, because you got to start thinking about homologous recombination versus replication restart in order to fit all these models in. But the take-home message here is that it definitely behaves differently on a junction with a 3' overhang versus a 5' overhang. So I'm happy to talk about all the data, after the talk's over. Okay, so in summary, I've showed you many different pieces over here. Like I said, it is a complicated protein with respect to the biophysics, the way it actually moves with respect to the dynamic structures. If you try to solve cryo-EM structures of RPA, which we have tried with many different complexes, every single time you see the trimerization core, you will not see the other domains of RPA. So, and so we have to rely on sort of very sophisticated experiments, biophysical experiments, and single molecule experiments to understand how the domains of RPA are used in biology. As I said, we've touched on many different parts over here. So RPA, the different bits of it, the trimerization code is less dynamic. The AB, the high affinity domains, I don't like calling them high affinity domains anymore. I'm just going to call them dynamic versus less dynamic halves of RPA. So I just showed you that we should be considering maybe DNA being more wrapped around RPA, and that would then alter the diffusion properties of RPA that I just showed you. I also showed you how different phosphorylations are changing the way the configurations of the domains of RPA are changed, and they have different roles in mitosis versus other parts of the cell cycle. I showed you how East RPA can be regulated by this protein called RTD105, which holds the domains together, which then prevents it from binding to interacting proteins, which are then licensed by DNA to open it up. So there's clearly an order to the progression of events and how these proteins are regulated and interactions are triggered. We've now started looking at binding of other proteins in DNA repair, and of course helicases sort of moving around and pushing them on the DNA. So with that, I think I will stop, and I'll just give my thanks to all the collaborators and the people in my lab who've sort of done the work over the many years, and funding from the Department of Energy to buy the EIFlex, and then IGMS for supporting the work a long time. So again, if anybody's interested in postdocs or being a grad student or a scientist and want to play with all the toys that we got, please come see us in St. Louis. We have definitely moved a long way since this movie was made, so it's definitely a much nicer city now. So And so, yeah, I'll stop there and open it up for questions.
Fabulous talk, Edwin. Thank you so much.